Since most of you, or many of you won't have heard of Anglia Ruskin, I thought I'd start off with two quick slides. Just here. We have a postgraduate campus at Chelmsford, which is in Essex. It's got a new campus, a very nice campus, and we have a very nicely equipped research laboratory, which is essentially set out to do lots of PCRs. Now, I think most of you are familiar with real-time quantitative PCR, and I don't want to go into any details, except to point out the fact that this is the usual format it is done in, and you have a single, my thing doesn't work. You have a single well in which you perform your assay. So typically what you do is you fill this well with your sample, you run a standard curve, and because you know the size of the pathogen genome, and you know how much DNA you had in the first place, you kind of work out a standard curve, and that allows you quantification of your target. I think you all know about this. Now, there's a problem with this, and that is people don't tend to propagate their errors when in their calculations. And so here we have an example of different aspergilli and the most commonly used target, which is ribosomal RNA here. And so we know that a single Aspergillus fumigatus, for example, has roughly 50 copies of ribosomal RNA DNA. So what you do is you do a quantification of Aspergillus fumigatus, and you have your nice replicates here for your qPCR, which is typical. As you have less and less target, your replicates get slightly worse, but even so, you can get a standard curve. And importantly, you really need to propagate your error, as I've done here. Most people don't do that. And so you end up with a calculation of a range, a genome copies and the range, and the actual targets. So I'm getting confused with my different gadgets here. Is listed then here, the ribosomal RNA range. Of course, most people will only show you this number here, and they won't actually show you the error that's involved. And as you can see, it's a huge error. So it is not terribly quantitative. And that is the problem with qPCR, not just for Aspergillus, for lots of different things. You draw your results, and you can see you can't really quantify this terribly well at the lowest concentrations. And even at the higher concentrations, there's a lot of overlap if you do your error propagation correctly. So qPCR is useful, but it's problematic. A new technique is called digital PCR, and it it will help avoid some of the problems. Now, digital PCR this is a digital PCR plate, again 96 wells, but instead of having 96 individual reactions, each of these wells now is split into lots of different compartments, as you can see here. And if you blow it up, you can see that instead of the PCR in a single tube, or a vessel, you do the PCRs in lots of individual reactions. And the workflow is very simple. 5-minute priming of your plate, 5 minutes dispensing your plate, 50 minutes priming, your PCR, and then you read it. So it's less than half an hour usually. So what you do, well, you just add your sample, and what happens is that the single sample is now distributed amongst X number of partitions. In some cases, there's lots of partitions. For example, the Bio-Rad instrument gives you roughly 18,000 partitions. Here we have about 500 partitions. The dynamic range of the assay depends on how many partitions you have. And you dilute your sample, essentially, and the idea is to have a single PCR in each partition. And this is what you get here. And then you use statistics to work out the copy numbers you have. In fact, just counting them. That's the easiest way of doing it. And so here's a use for this sort of application. Here we have 3 different Aspergillus samples. They were quantified using a nanodrop at 40 femtograms. And we ran these on our digital PCR instrument. And as you can see, we can now count at the highest dilution. We actually count copy numbers of ribosomal DNA. If you compare this to qPCR, this is what it looks like. We have these 3 Aspergilli. These are the ribosomal RNA DNA repeats. So we have that many genomes, that's the amount we had. That's the copy numbers we expect to get. If we use qPCR, we get kind of the same result. Certainly within the error, we're getting the same result. But you can see it is not terribly accurate. But what's interesting is that the qPCR would suggest that the ribosomal DNA repeats for the terriers are not correct. When we do this using digital PCR, we get a similar result, except it's much more precise now. Now, this is a log scale here. On the qPCR, this isn't. And you can see we now end up with a copy number of 95, 46, and 30 with a very, very small range. So digital PCR is extremely useful for accurate and precise quantification of DNA. So this might be useful, for example, when you're producing your samples for shipping out to different people, your panels, so you know exactly how much DNA you have there. The proximity ligation assay, which Gemma talked to you about briefly, we've used two different methods. There's two main, the main instrument on the market is Bio-Rad's a QX 100 or 200. The problem with that instrument is twofold. Firstly, you have to use their reagents, which makes it expensive. And secondly, because you're using proteins and the Bio-Rad instrument depends on droplet generation, it interferes with the droplets and you don't get a lot of droplets. So instead of getting 18,000 droplets, you end up getting 12,000 droplets. And so you have a problem with the quality of your results. I'm switching slightly now because we're interested in another pathogen, which is Clostridium difficile, and I'm just going to show you some data on that. This is the problem that Gemma alluded to. If you do a PCR-based PLA, it's very difficult to distinguish your no protein control from your very, very highest dilutions. So you can see these are simply repeat PLAs. That's the wrong one. You can see it is very difficult to see what's happening there. Sometimes you get a nice difference, sometimes you don't. Now, digital PCR takes care of that, and here's the result. Here you can see the actual data, and here's the interpretation. You can see you get a very, very clear distinction between the lowest level of C. diff antigen and the NPC. In fact, we're beginning to eliminate the problem of background. So this is something we're working on at the moment. Now, with Aspergillus, we're not quite as far yet. This is similar to what Gemma showed you, and we're simply comparing a PLA on these two concentrations or dilutions of Aspergillus using a qPCR-based assay and using a digital PCR-based assay. And you can see that there is still scope for improvement on the digital PCR, where there probably isn't much scope for improvement on the PCR-based system, qPCR-based system. And here is the data. qPCR, big error bars, not really possible to say what's what, whereas digital PCR, very nice, precise separation of the different amounts of target DNA, or protein in this case. So where's the future? Well, I'll be controversial now. I feel slightly out of my depth because I'm not a mycologist, I'm a molecular biologist, but I think it is clear that there are problems with the assays that you are using. And the future of galactomannan, well, I don't know, but clearly we and others have identified problems with the assay. The problem with PCR is that it is not a robust assay. It is not different people get different results. And here's a very nice study, which is, I think, one of your studies. And to me, as a PCR person, and this is not just limited to your study, this is typical of what you find when people do comparative PCR analyses. There's a huge range of results of the sensitivity that different labs get. And in this case, if you actually plot the data, there's a 1,000-fold difference between the best and the worst lab. And one of the labs didn't get any results at all. So I think you just discussed the inclusion of PCR criteria into your. I think you have a real problem there, because until you've managed to get people to get the same assay, the same levels of sensitivity, you will still have the problems they have today. And I think someone needs to really look at which assay to use and how to use it properly. I think this is a very nice way forward. We have, as we've already heard, the lateral flow device, which is very easy to use and very specific, and we can combine that with a PLA, which uses the same antibody, which gives it the extra bit of sensitivity that, if you remember Chris's talk, would bring it up to the level of PCR that has been identified in your study. I think qPCR is going to be very useful for species identification, because it's obviously going to be able to distinguish individual DNA sequences. And I think digital PCR will be very useful for monitoring patients because it allows you to very accurately quantify the fungal load. Right, so in summary, sorry, this is a translation from Mac, so something's happened here. But in summary, we have PLA, LFD, early quantification detection, multiplex qPCR, species detection, and then digital PCR for monitoring. And I think we are well on our way towards on a path that allows us the early identification of fungal infections. But I think a combination of different techniques, in my opinion, it'll be PLA, PCR, digital PCR, and qPCR that'll do all of that. And I think that's it here. Thank you very much.